high, love. Raising high, love.
So I'm an engineer. I have my degrees in both engineering and science. And I think it's the very best time to be an engineer right now. It's an exciting time, and there's so many opportunities to make an impact in the world. And so what I wanted to do was to talk to you some about what we do and convince you here today that you're our future in science and engineering, and convince you to join us in trying to solve some of the world's biggest problems. Some of the problems that we think are the problems the most pressing to the planet are food, how to get enough food for everyone, how to get water, energy, solar energy, batteries, and medicine. And so in my group we study actually all of these different problems, and we solve them by looking at two things. One is nanoscience, which I'll tell you a little bit about, and the other one is biology. And so we look at this interface between nanoscience and biology. So people said that, I heard over here that that nanoscience or nanotechnology meant a billionth of something. So is that very, very big or very, very small? It's very, very small. And so how many people have seen a nano iPod before? Have you seen those? Okay, so they're definitely really, really cool, and people think that what's so great about nano is that it's small, and it's great to be small. You can have smaller phones and games, but the other thing that's so great about nano is when you only have a couple of atoms, and when you take a couple of atoms and you control their size, it can have really great properties of things like solar cells and batteries and water purification and medicine. And so what we're interested in doing is controlling atoms at the nanoscale. So our inspiration comes from abalone shells, like you see there, and the reason that we're so interested in them is because that shell that you have, or that I passed around, it has all the information on how to grow it inside of its cells, inside of its DNA. And so the DNA says, let's build this really beautiful structure. Another inspiration are trees. Anyone know anything special about trees? How they get there? Yeah, what do you think is special about trees? They're durable? Is that what you said? Okay. What do you think? Yeah, they do. They help with pollution? Yes. Do you want to come work in my lab? Yeah, that's right. So all those were really great comments. Yes, they use photosynthesis to get their energy. They take sunlight and they use it for their energy source. Basically, when two abalone get together, a male and female abalone get together, they make lots of little baby abalone, and they pass on to their baby abalone the DNA that said this is how to build a beautiful structure of calcium carbonate. So what we've done in my lab is say, what if you did that with a solar cell or a battery? What if a solar cell said, this is a way to pass on the information and make lots more solar cells? Or what if it passed on the genetic information and said, let's make lots of batteries. Let's have a DNA sequence that says this is how to code for a battery. And that's basically what we've worked on. These are actually little super antennas. You can come see them afterwards that are really great at absorbing light for examples like photosynthesis that we've genetically engineered. We've manipulated their DNA so that they're great at collecting light for solar cells and other kinds of applications. But the example on the bottom are called diatoms. Anyone ever heard of a diatom before? Diatoms are made out of silica. They're made out of glass. Organisms in the ocean can make glass at ocean temperatures. Can you imagine that? You've used glasses to drink out all the time. They're little bitty organisms that make beautiful glass structures, and they have the DNA sequence that says this is how to make them. Now this is one of my sons, I call him my favorite biomaterial. He's standing next to his favorite biomaterial. Do you guys know what that is? The Triceratops. But anyone that has little brothers or sisters in the audience know that little kids are incredibly complicated organisms, aren't they? Aren't your little brothers and sisters incredibly complicated organisms? Yes. And so they're really hard to control. I can't even get my 3-year-old to put on his shoes. And so it'd be really hard to get him to grow a battery. And so instead of using really complicated structures, we use things like bacteria and viruses. So here's some small things in biology. I'm going to see how many of these biomolecules you have heard of. How many people have heard of DNA? Yep. DNA is a genetic code that codes for all the functions in our body. What about antibodies? Anyone heard of antibodies? Antibodies are proteins. They're part of the immune system that can actually grab onto things in the body. And there's other things that are called proteins. Now, one of the things that's so nice about these is they're already really, really small. So do you guys understand that you're all nanostructured materials sitting here today? You're all based on molecules and nanoscience. So our idea was, can we take these molecules and convince them not to do their normal things, but to convince them to grow solar cells and other structures? Here's some pictures of some natural biomaterials. That's the abalone shell. Here's the secret to why that abalone shell is so strong and so tough. If you look at that picture right there that looks like a brick wall, that's a brick wall on the nanoscale. Basically, the abalone decided to make its calcium carbonate that's like a brick wall structure, so it's very, very hard to break. And that actually has, the other secret to it, besides it being this nanoscaled brick wall, is that it's 2% protein, and that protein acts like a glue and sticks those structures together. The one that looks like a spaceship at the bottom, that's a diatom that's made out of silica. If you take an abalone and you look at it in a scanning electron micrograph, anyone ever seen an electron microscope before? Some people in the audience have seen electron microscopes before. It's a way of looking at really, really small things at the nanoscale. If you look at an abalone at the nanoscale, what you'll see are these little tablets, and in between each little tablet is a little bit of protein, and that protein acts like a glue. Anyone have pearl earrings on, or a pearl necklace? Those are nanomaterials. These are abalone shells, or actually oysters that make these structures. So the pearls are actually the same, have the same properties of shells that these nanostructured brick wall-like structures. So here's the periodic table that I handed out. Now, biology in the oceans only used a couple of elements. It's used calcium. You can find it later. It's used silicon to make diatoms. It uses phosphorus to make bones, calcium phosphate. But most of the periodic table, biology doesn't use to make materials. And we said, that's too bad. What if biology could use the whole periodic table? What if it could use all those other elements? And the reason it hasn't used all those other elements is because it hasn't had the opportunity through evolution. Anyone ever hear of Darwin before? Anyone hear of Darwinian process of selection, the survival of the fittest? What we do in my lab is we say, let's use the Darwinian process, let's use the survival of the fittest process to find a little organism like a bacteria or a virus or a yeast that can grow an electronic material that it wasn't meant to grow through evolution. So here's again part of the secret of how the abalone makes that shell that I passed around. It has proteins, right? And proteins are made out of what? They're made out of amino acids, and those amino acids have different chemical functionalities. Well, the proteins that make up the abalone shell are very negative. They have a lot of negative charge, and they grab the calcium ions out of the environment. They build up calcium carbonate, calcium carbonate, calcium carbonate, till they form this brick wall-like structure. It was about 500 million years ago that organisms started making things like shells and diatoms, but it took them 50 million years. How many people plan to go to college for 50 million years? Anyone here? No, it's going to be really expensive. What, you're going to go? You'll be really smart when you get out. So it takes a really, really long time. So what my group decided to do, what my researchers decided to do, was to make a shortcut. Instead of taking 50 million years to build a battery, we decided to do 1 billion experiments at a time. The trick is that we went in and we changed the genetic information of a virus to add a really small amount of extra DNA. And so we used little molecular scissors to cut the DNA. Anyone know what kind of molecular scissors you can use to cut DNA? I saw that hand back there. Yep. Restriction enzymes. All right. Yeah, you're really advanced too. So restriction enzymes are little enzymes, little proteins that can cut DNA in particular places. So we use restriction enzymes to cut the DNA and open it up and put new DNA into it. And so basically we do that 1 billion times. So you have 1 billion viruses that are all genetically identical, but they differ from each other based on a small amount of amino acids on the other side of that periodic table. Now you take 1 billion viruses and you force them to interact with a battery material. Most of them don't want to interact with it, but maybe 1,000 out of 1 billion will. And you say, okay, those 1,000, this is the Darwinian process, that 1,000 out of 1 billion will interact. Well, now you say, now I'm going to make it harder for you to interact. So you find 1 out of 1 billion that can do what you want to do, like to grow a battery. And here's a cartoon that some of my students made about one of my collaborators and myself on things that we've been able to do with viruses. We've been able to build solar cells and batteries. We've been able to make new kinds of materials that can convert natural gas into gasoline. We've been able to engineer yeast that are the same kind of yeast that make beer or bread. Instead of making bread, can convert CO2 from a coal factory and make it into tiles, green tiles that you can walk on for floors. We've also developed DNA sequences that based on viruses that can find very tiny tumors inside the body, sit there and give a signal so that surgeons going in and doing surgery on ovarian cancer patients can find tiny tumors and locate them and be able to remove them. And then we've also made viruses that can take sunlight and convert water into oxygen and hydrogen that can be used for fuel, and we have some over here. So all of this has been done by manipulating the sequences of these organisms. So these little wire-like structures? These are wires that are no longer really biological, but have taken ions or elements from solution and built these continuous wires. Now we can take these wires and make millions and billions of them, and that's the components that make up these batteries that I showed you today. So this is part of my students wanting to make this battery for the car. So this is a virus that we genetically engineered. It's stable in water, so you just put it in water in solution. And we're able to give it ions, and it's able to grow the structure called manganese oxide. We give it a little bit more genetic information, and it's able to add a second element to form these wires. Now these wires are going to act as batteries to take lithium and oxygen to use as a battery for long-distance vehicle applications. And through this ability to manipulate the DNA and genetically engineer it, we made this battery that was a record-based battery at the time. You take lots of them and they self-assemble to form structures, and then you can actually use them in larger batteries. And one day we hope to be able to use them for long-distance vehicle applications. So one day you may have a battery in your vehicle. This is a nice vehicle that will be based on biological organisms and will be environmentally friendly, and you'll be able to go longer distances. So with that, I'll leave it there.